So I'd like to welcome Cindy to the International Febby Series. It's a great pleasure to have her here because I've been trying to get into South Africa for years. I might be lucky. She may be coming in May, which will be great. So those of you that attended ICPP will remember that Cindy gave one of the plenary lectures at the conference. And then she gave the first lecture at a concurrent session I organized on bacterial taxonomy. And it's the first time in my life, in my career, that I've attended a session on taxonomy where there's been standing room only. So you can imagine what a very good speaker she is. So her speciality is on Pseudomonas syringae and mainly the water cycles. She's an epidemiologist, ecologist, and more importantly, and I found this quite amazing, is that she composes her own songs and performs them. And those of you that can remember, Mike and I shared her videos during COVID, and she sang about COVID. Those of you who would like to hear those videos, I think I still have them on your email. And with that, I'd like to invite Cindy to give the seminar.
Okay, well, thank you. You can all hear me fine? Thank you very much for the invitation. And someday I'm getting there before the end of my career. I'm going to South Africa. Maybe it will be in September. We'll see. But anyway, I want to come so badly. So thank you very much for this invitation. I'm going to share my slides here so I can get started. I want to talk to you today about how to approach anticipating plant disease. And I'm going to use the model of Pseudomonas syringae because it's so complicated. And that's the title. This got pervasive reservoirs, long distance aerial spread, variable host range, and all kinds of things that make it really complicated to anticipate. But it's not the only one. So when I'm talking, you can think about your own models or models you know and how this might be applicable. So in Europe, there's been many initiatives to reduce pesticide use in agriculture, and this has created lots of opportunities for research. So we've had a European directive that started in 2009, and this led to some goals by the European Union to reduce pesticides. And finally, France opened a priority funding program to think about how to move toward zero pesticides in agriculture. So this main goal is, part of the main goal is to improve surveillance and disease anticipation as one of the ways to reduce pesticides, and this would foster prophylaxis, biocontrol, and agroecology. So I'm a beneficiary of this funding, and I'm just right in it. And so now my thinking is really going on around this idea of surveillance. So this complexity of Pseudomonas syringae's ecology and the epidemiology of the diseases it causes really motivates questions about how to improve disease anticipation when things are difficult. So for those of you who know a little bit about the model of Pseudomonas syringae, we all know it's a plant pathogen. It causes disease to many, many different crops. But my work has been around the fact that it's in the water cycle. And then when you find it in clouds and rain and snow and rivers and everything, then you ask the question, are they pathogenic? And this led us to doing host range testing, which showed how broad the host range is. And so you really have questions about how to survey this big spectrum of habitats where it can be. So the questions that we have pursued to expand the scope of disease surveillance are the following. So can we survey air and water, but especially for the long distance dissemination, not just the short distance dissemination, but long distance? And what will be the surveillance targets and what tools should we use? Okay, and then can these approaches for surveying air and water be transferred to other pathogens? And then how will this open opportunities for prophylaxis in anticipation of disease? And so this has been, I've been able to do this with funding from two major projects that actually probably the only funding I ever got in my research life. But anyway, at the end of my career, it happens. It's really, since 2018, we're comfortable and the dynamic has really been fun. But the last one is building epidemiological surveillance and prophylaxis with observations that are near, but also distant and maybe not as obviously relevant, but we need to find other indicators. So we have 4 areas that we're focusing on in the work on Pseudomonas syringae and in this project. So if we want to look at dissemination via surface water, we want to know what's the extent that a pathogen can be extended in a watershed. Is it local or is it spread everywhere? Then if we're looking at dissemination via the atmosphere, what's the scale and what are the trajectories that it could take? Then we have questions about what are reservoirs of potential pathogens? And then finally, with all that information, what should we be watching for in surveillance? Okay, what and where? Okay, it's a complicated question. So I'm going to go through each of these points. So the first question is about the extent that Pseudomonas syringae is present in a watershed. And so the question we asked is Pseudomonas syringae present throughout the full extent of a river basin or only in some locations, like, for example, near diseased crops? All right. And so we focused our work on the Durance River Basin, which is in southeastern France. This river basin is in a part of France where there's 27% of France's horticultural crop producers and 21% of its fruit orchardists are here. So it's really the fruit and vegetable basket of France. And so we went and we identified 21 sites along nearly the entire reach of the river. So 300 kilometers, we did 270 kilometers of the reach of the river. Now the little dots on this map, it shows you the river. And the altitude spanned from 39 meters to over 2,000 meters. And we took samples in 2 years at 4 different seasons. So we have 8 samples, 2 in winter, 2 spring, 2 summer, and 2 fall. And so we just took water and we were measuring the amount of Pseudomonas syringae, and then we checked its diversity. So from that study, of course, we do direct isolation because we want to be able to do pathogenicity testing and other characterization. We isolated over 5,000 isolates of putative Pseudomonas syringae. So those were the ones that we were then going to say, are these Pseudomonas syringae? And then we were able to attribute the phylogroup of 2,628 of them as Pseudomonas syringae via CTS sequencing. So that's the sequence of it's a 388 base pair segment of the citrate synthase gene that is really useful in identification. And so, but we also had water chemistry and temperature information. And we did this work in collaboration with Marianne Barney and Frederic van Giesegem, because in parallel, they looked at the soft rot enteric. So I'm not going to talk too much about the soft rot enterics today, but we did a comparative study with the same samples. And really, it was Marianne who really organized all the sampling. It was wonderful. So for Pseudomonas syringae, here's the basin, and these little bar graphs are for every site, the 8 different samples, 2 in winter, 2 in spring, 2 in summer, and 2 in fall. The y-axis goes from 10 to 10 to the 6th CFU per bacteria per liter. And basically, Pseudomonas syringae is everywhere all the time. So this is sort of like a complicated situation. So it's present at all sites and in 155 of the 168 samples we took. This is not the same for the soft rot enterics, and you can read about those details in a paper, but Pseudomonas syringae is there all the time, all right? And also, we looked at all the correlations with the environmental parameters, and really temperature was the most important explanatory factor. In this graph on the left, you see that Pseudomonas syringae, the values for Pseudomonas syringae relative to water temperature are the solid dots, and the ones for the soft rot enterics are the open dots, and they have absolutely inverse correlations. Okay, so Pseudomonas syringae population densities were inversely correlated with temperature, whereas the soft rot enterics were positively correlated with the temperature. And I mean, this is basically basic ecology that we know about these bacteria, but it really overrides everything. It has, for Pseudomonas syringae, has almost nothing to do with land use along the river. Okay, it really has to do with temperature. And so if you want to read the details, I'm just going to give a plug for this new journal system in France, this peer community journal, which is a very open access, no publication cost. All the participants participate in all the steps of the editorial process and reviewing. And that's where we published it. So it's you can find that on internet. And so now that's for the river water. Okay. The next step is atmosphere. If we want to study the atmosphere, how do we approach this? So we know that Pseudomonas syringae is transported to and falls with precipitation at remote sites. So we know that from collecting rain and snow that Pseudomonas syringae falls with the precipitation, but very, very far. And the most incredible collection we did of Pseudomonas syringae was at the Jungfraujoch site in Switzerland, which is at 3,580 meters of altitude. And this place, it's an observatory. It looks like Disneyland. It's so beautiful. It's an observatory that you can get to. It's the highest place in Europe that you can get to with public transportation. All right, you can get up there with a train, with 4 trains. You have to take 4 trains to get up there. And so I had a student who was working on a question about ice nucleation in the atmosphere, and he was collecting snow, and we did the microbiology of the snow. And on a couple occasions, we actually found Pseudomonas syringae in the snow. And so on one date in particular, on 22nd of October, he found 45 cells per liter of snow melt. So, okay, I said, okay, what's diversity? So just ask yourself, what would you expect? One snowfall event, what kind of diversity of any pathogen would you expect? All homogeneous, diverse? I had no idea what to expect, but I was shocked when I saw this. So this is the phylogenetic tree of Pseudomonas syringae, roughly, all the phylogroups, and the dots represent strains from this snowfall sample. The other strains are in there to give you the full spectrum of the diversity of Pseudomonas syringae. So I just said, like, wow, okay, that was crazy. So that really raised the question about where the snow got that, where the air mass got those syringae from to drop them out as snowfall. We'll eventually talk about this in this talk, but high diversity of Pseudomonas syringae is transported a very, very long way to places that are irrelevant for agriculture, okay? When this snow fell, the whole place was covered with snow, so it was not a local source. So we're going to continue looking at long distance transportation for a while. So another thing we noticed is that with the availability of lots of whole genomes, I started to get curious about lines of Pseudomonas syringae that were in these haplotypes of CTS that I started finding in many places. And so by looking at the genomes that were available from my own work and other people's work, we found that there was a quasi-clone of strains in phylogroup 2D that is present across France in crops and other substrates and is also transported in the atmosphere. So when I say quasi-clones, they were identical for about 1,500 to 1,800 loci out of the 1,900 loci in the core genome. And these were found at 14 locations at a whole bunch of different years. And so here's where they fell out. This is the example, this quasi-clone. You have a bunch of them that were falling out we found in areas where there were cantaloupe blights and cantaloupe culture, but we also found them in a local irrigation basin. But then in eastern France, we found them with snowfall, irrigation water, biofilms, groundwater, rainfall, where there's not the disease. There's cantaloupe cultivation, but there's very little disease because of the climate conditions. So this is extremely interesting. So this is basically telling you that this organism is moving around to all these different places. Then we found another clonal line, so what we'd call a clonal complex, technically, by the people who study clones, that's in crops, and this is from reports from other people, in crops on 2 continents in the northern hemisphere and in a creek in a remote setting in the southern hemisphere. And these are 99.68% similar based on their whole genome. And I mean, especially for the one in the southern hemisphere where it was in a pristine place where there's no agriculture, this is likely due to dissemination via the atmosphere. And so if you want to see a picture of that, these are the different places where this clonal complex was found at different dates in France on zucchini and cantaloupe, in Serbia, in Florida, and in New Zealand in the outback. And that was collected by Joel Van Est. And I mean, it's just it tells you about how much dissemination can happen with this organism. All right, so we know that Pseudomonas syringae is frequently detected in precipitation. So one question is, can we observe it directly moving from one place to another? How would we do that? Okay, so that's one option to do. Can we trace its movement via direct observations from rainfall or snowfall along a storm? Okay, so we tried to do this. Very complicated. So thanks to the participation of my colleagues at at the lab who live in quite a few different places, not right next to where we work. Okay, they live right like about in a 30-kilometer radius. And they were willing to set up rain samplers in their home. And so there were 13 participants at 14 sites, and I set up a WhatsApp warning group to tell them, tonight you collect rain, or this morning you collect rain. And they had all the materials and the procedure, and they would bring the rain samples in. And then I would do the microbiological analysis, and then we'd look to see with the weather mapping if the samples were connected, if the air masses had moved from one place to another. So I was able to get 3 sets of connected samples, which was not much for a lot of work, but that's how it is. I mean, some people collect the rain and then they can't bring it in on time, or some people didn't get up in time to collect the rain, or their bag broke or whatever. So these are 2 examples that I'm going to show you about what we tried to do. So the yellow dots on these maps are where we collected samples that had Pseudomonas syringae in them. And then the red and yellow lines are the information about if there were air masses moving between those places. So on the left, on March 2nd, we see there's 2 sites that the air masses, the storm was moving from to the west. Okay, the storm was moving to the west. And then there's a storm, there's air going up to the north. Okay. And then on June 4th, there were some sites that were also connected, okay? And so what I want to look at is the genetic distance between these samples. It should be smaller than the genetic distance of all the different syringe strains than between samples that aren't connected, okay? So that was the hypothesis. So if they're being moved, all right? So for syringe populations that are disseminated by the air, we would expect the genetic distance between sites connected by air movement to be less than between unconnected sites, if air movement is really the thing that's happening. So here are the results. I compared genetic distance within a sample, between connected samples, and between unconnected samples. So no significant differences, but you can see here the trend is not at all what we would hope for. Okay, we have less genetic distance between non-connected samples, but look at the amount of genetic diversity just within a single sample. It is huge. And so it just means this hypothesis is extremely difficult to test unless you have a huge sample size within a lot of strains within each sample. Likewise, for the second time, this trend is what we were hoping for, but there's so much genetic variability that we can't see anything significant. So it was very, very shocking how much genetic variability. If you just take a rain sample, how many different Pseudomonas syringae are in one rain sample? It's absolutely mind-blowing. So this was a difficult thing to do, and I wouldn't recommend it for a young scientist who's trying to get their first publication. This is for the old people to can take risks, okay? All right, so then there's another way to go about this, okay? Maybe the presence of Pseudomonas syringae in precipitation depends on the geographic trajectories of air masses. So we can not necessarily see them, but we can say, well, they came from here, so we expect them to be more or less abundant. And this was something that was done within the thesis of my graduate student, Caroline Montoy. So on the left, we have mapped all of the different, we collected things mostly in France, right? And all of those dots or trajectories that arrived at the sampling site. So we have air masses that come from many, many places in the 88 samples that we took. And we measured conductivity and pH. And so already we could see that there was the pH of and the conductivity were higher when Pseudomonas syringae was present than when it was absent. So that tells you something about its association. The air mass, when you have high conductivity, you generally have water coming from the seas with salt in it, okay? And so that tells you something about the air masses that are coming from the west, okay, or the southwest, mostly have, and a little bit from the northwest, but not much, would more likely carry Pseudomonas syringae. And likewise, on the right, the graph on the right, we have the geographic origin of all samples in red. So most of them are from the north and northeast. Some are from the west, and a few are from the south. But if we look at the frequency that have with Pseudomonas syringae in them, most are from the south. okay, and a little bit from the west, and very few from the north. So this is giving you signals that there are geographic origins of the Pseudomonas syringae that's arriving at any given place, right? And so then the next question, we said, can we predict the arrival of air masses that are likely to disseminate Pseudomonas syringae? So because we'd want to set up sampling intelligently rather than just running around with our samplers like, you know, crazy people running around, but we want to, it's a lot of work, this sampling in the atmosphere. And so where should we put up our samples and when? Samplers and when? So all these trajectory data that I'm showing you, these are things that are obtainable through the National Oceanographic Atmosphere Administrative Site run by the US, so NOAA, okay? And you can calculate, if it rained yesterday at your place, you could say, oh, where did the air mass come from that brought that rainfall? And if you put into the high split model, the geographic location and the date, it will tell you where the air masses came from. So this is a modeling, the Lagrangian modeling that it does. So we had a doctoral student in 2020, Maria Shufeni, that basically we tried to look if we could calculate the reliability of these atmospheric networks, whether any patterns in them. So first thing she did, see, these are statisticians, these are not biologists who can do this, all right? So she compiled data on air mass trajectories between given sites across a geographic region. So we focused on our region of France. She used this archive data and she downloaded and compiled 10 million trajectories that occurred between 2011 and 2017 from the NOAA data bank, the high split site. Okay, so she did that, and then she used that. So this map, can you see the animation, Teresa? Just put your thumb up if you can see the animation. Yes, okay. So this is the PACA region, so our region of France. Each dot in the middle is the center point of where she calculated. She said, okay, I want the connectivity between that point and another point. These are all, would be these 290 areas that she delimited are topographical watersheds, okay? And then she said, show me, based on all these air mass movements, what sites are connected to each other. So in the black, are sites that stay connected over a longer period of time. So in addition to those little lines you see moving, you see the date, 2013, May 4th, May 5th, or April 4th, April 7th, 9th, 10th, as the days changes. So you can see during seasons, the connectivity of these sites changes, direction, intensity, okay? But if she would do this same pattern for 2014, it would look the same. It's really a seasonal effect. It's not a yearly effect, all right? So this was part of her work, all right? And so from that, we created a tool that we could automate the calculations of these multiple trajectories, because most people are not capable of downloading 10 million trajectories. And for your own work, you might only need a couple thousand trajectories. So we created a tool that now automates this. And that's the tool is described in this paper. And if you want to use the tool called TropoLink, you just have to write to me and I'll get you in line with the person who can open it up for you. You can then just easily tell TropoLink, these are my geographic sites I'm interested in if they're connected via air mass. These are the periods of time I want to look at. And then please calculate for me the connectivity. So that's the network analysis that statisticians do, and it will show you what are the highly connected sites and how it changes over time. And so this now is a very generic tool, okay, that we could use, for example, to identify seasonal sources and sinks for air mass movement. So for example, here's a map that the colleagues made that based on those data, they say what sites are linked in the winter and summer, okay, and what's a source and what's a sink. So you can see in winter the sources are indicated in red and the sinks are indicated in blue. That would tell you that things are generally moving from those red places to the blue places. And likewise for summer, the sources and sink change, and makes sense because crop production changes, right? And also seasons change, right? So although we were asking questions for Pseudomonas syringae, all microbes that are transmitted long distance in the air are moved passively. They depend on these atmospheric highways. So this is a tool that could be used for developing surveillance procedures for any airly disseminated pathogen or small insect. You overlay this with land use, and then you implement a coupled surveillance of target organisms in your source and your potential sinks. And you say, oh, now I collect my populations in my source and my potential, or my sink, you know your sink, but your source, my potential source, and then you can do the genetic comparisons to see if they're comparable. Okay, so that would allow you to validate the transmission. Okay, but then we looked at other applications where you can enhance the pertinence of the trajectories that you estimate by taking out times of year that aren't pertinent. So if you have a certain microorganism that's never available in winter, you wouldn't use those winter trajectories. Or the plant phenology stages that wouldn't be emitting spores and bacteria, you would take those out too. So you would make your trajectories more pertinent. And then you could create a network, and you can, via network analysis and calculation of these hubs, the points that connect all the different lines, you could say, what's my optimal positioning of surveillance? So for example, how many sites, how many hubs do I have to put monitors of surveillance to have information about 90% of the network without surveilling 90% of the network? This is what network analysis does. So I encourage you to read this paper. André Radici, who is the author of this paper, is a graduate student in our Beyond program who's looking at this. And then if you think about diseases that are disseminated across Europe, like wheat rust, there's a lot of countries that are involved. And so sometimes the countries could say, well, maybe I can just not have to survey because I don't have any hubs in my country, and I can depend on other countries. Can we do cooperation? What would be the trade-off? Would there be any financial gains? Would I be at a risk of losing information for an outbreak locally? So you can also look at political trade-offs for how to do these surveillance things cooperatively. And again, this is by Andrea. It's a extremely interesting paper about how you can optimize this cooperative surveillance. So this started out with Pseudomonas syringae, but now these tools are pertinent for mapping any potential, the highways that microorganisms can ride on. All right, so now the other question we needed to ask: are there geographical limits to the aerial dissemination of Pseudomonas syringae? So now we know it's in the area. I've taken you up to Jungfrau Joch. You think, oh, I can go anywhere now. Are there limits? What would be the limit? We need to know that. All right, so we'll go back to Jungfrau Joch. And my question was, where did that air mass come from? And so we used the backward trajectory measurements of the meteorological station. So that's this green and purple blob on the map. So the map indicates where the air mass came from to arrive at Jungfrau Joch, and the greener the color, the closer to the ground the air mass got. Okay, so the more purple color is very high altitude. So I was interested in air masses that were kind of scrubbing the ground before they arrived to Jungfrau Joch. And so among the different sites, there was one possible source that I thought would be way too cool to go to, and that's Iceland. Right? No agriculture in Iceland. 1% of it, land use is agriculture. I thought, wow, that would be a real extreme case of movement. All right, dissemination. That would be really, really interesting. So off we went. We went to Iceland. We got funding from a Campus France Jules Verne program, and then eventually from the Icelandic Research Foundation. And we sampled for 3 years. My job was to sample on monocots, dicots, fern, moss, and litter. We eventually had a graduate student that went to lichens, and I'll let her eventually tell that story, but we also found them on some lichens. So this story is just about angiosperms, fern, moss, and litter. So we had 16 sites all over, well, not all over Iceland. What's important is we weren't in the very, very far south, but we were mostly, because there's a lot of glaciers and it's more touristy, and it's complicated when you go there. It's easy, but it's too easy because there's a lot of people. So we had 16 sites and 38 samples. And so, yes, success. We found them on monocots, dicots, moss, fern, and litter. So here are just the population sizes on average of what we found on the different monocots we sampled, on the dicots, on moss, and then fern and litter, we only had one sample. So Pseudomonas syringae is the triangle, and the total populations are in the circles. All right, so we found them on almost everything we sampled. But the genetic diversity was not at all what we expected. Okay, because what's the hypothesis? The hypothesis is that they're coming, the ones on Jungfrau Joch maybe are coming from Iceland. So we would expect the genetic diversity to fall right within the map of Pseudomonas syringae. We'd expect to find similar haplotypes. That's not what we found at all. I was shocked. There's not one haplotype that we found in Iceland that we found anywhere else in the world. And we have a database of like 900 strains, 900 haplotypes. So the Pseudomonas syringae haplotypes, CTS haplotypes, so we're talking about 388 base pairs, all right, are unique to Iceland. And furthermore, they make monophyletic groups. It's not just like here and there. They're branched off, all right? And they're distinct. Whereas I showed you, there's haplotypes that we find all over the world, elsewhere. But these haplotypes, they weren't like anything else we saw. So I went, oh boy, I don't get this. I don't understand. So this is a publication. You can read about it, and I will explain to you later on why this happened. But we're going to go on to another story first, because of course you want to know, well, what does this mean? And I will explain how this is coherent with dissemination, but we'll go on to another subject first. Okay, so keeps you in suspense till the end. So let's look at the reservoirs of potential pathogens. This might be the story that many of you already know about, because I've made a lot of noise about this one. So this was one of the first questions we asked about Pseudomonas syringae. I couldn't believe that Pseudomonas syringae was only associated with crops. And when we started looking, we just found it everywhere. So this is a summary. This slide is a summary of all the different sources that we found it on and other people have found it on. So, and then the graph is log of bacteria, either per gram of tissue or meter squared of ground cover or per volume of water, okay, etc. So crops, healthy crops, wild plants, epilithic biofilms, leaf litter, cloud water, rain, snow, lakes, irrigation basins. We found it in groundwater at 7 meters below ground level and in aerosols. It's been found in aerosols. So of course, when we first published this, people said, "Oh, but those aren't pathogenic. Those are not the real Pseudomonas syringae." And that was. So I had to address that, and we did host range testing, and every substrate that we isolate Pseudomonas syringae from has lines of the bacterium that can cause disease in the same way that things we isolate from epidemics cause disease. So this is, of course, in the laboratory inoculation, wild alpine plants, leaf litter, snowpack, streams, lake, groundwater, cloud water, irrigation basins, and crop plants, of course, all have strains that can induce symptoms on the plants that we use, cantaloupe, beet, and lettuce. So these strains are like everything else. They're not anything special. And when we do the genetics, so this again is from the postdoc of Caroline Monte. She looked at whole genome sequences of strains that were from crops and environment. And when you make the tree, when you do the genealogies inferred by clonal frame, you can't, they're all mixed up. All right. If you pulled a strain out by random, you wouldn't be able to say this is an environmental strain or this is a crop strain. Okay. So they're all mixed up. So this is reinforcing the idea of mixing, but I've just been telling you that Pseudomonas syringae is flying everywhere. So this is totally coherent with what we have been seeing, okay, in the environment. So this brings us to the last question before we go back to Iceland. Don't forget, we're going back to Iceland. So what are the candidates for surveillance? So now if we want to synthesize all this into a question about surveillance. Should we survey only the clonal lines that have actually been declared in an epidemiology to have epidemiological impact? So even though I have strains from rivers and snowpack that can cause disease in the lab, should we only be surveying for the ones that have actually caused disease in crops? Okay, so that's a question. how could we then anticipate the emergence of new genetic lines if we didn't take into account the information that we have from environmental reservoirs? And then what types of Pseudomonas syringae pose the greatest risk? Okay, should we be surveying the ones that have the broadest host range, for example? All right, those are some questions we should ask for. So we go back to our strains in the river. We have 2,628 isolates of Pseudomonas syringae, and these represent 291 CTS haplotypes. So that's the diversity. That will tell you about the diversity that represent most of the known phylogroups of Pseudomonas syringae. Let's look at the frequency of those haplotypes. So this is a classic species abundance curve where you have a lot of haplotypes that were only detected once in all our sample. And then you have, that's there, about 40% of the strains represent ones that are only detected once in the sample. And then you have ones that were detected fewer and fewer times. And then you have one haplotype that constitutes 10% of all populations at all sites. So it's a CTS haplotype, 388 base pairs, okay? It's not necessarily a clone, right? But one haplotype, 10% of the sites. And this haplotype, we gave it a name, it's called DD1, and it's in this phylogroup 2B. And now I've already told you a little bit about 2B and 2D. These are the ones that are moving all over the place, okay? So again, it's not very surprising. So interestingly, this haplotype, if I go back to our big database of strains that we have from reports of, so sequences from reports of disease all over the world, this haplotype corresponds to reference strains that have been found in over 10 countries on, I think, 7 continents. Okay, so this haplotype is getting around and doing something bad, causing disease, right? Okay, so, and it's been also found from the widest diversity of habitats. In addition to crops, it's plants, rain, groundwater, rivers, etc. Okay, so is this a haplotype that is really adapted and has pathogenic potential? And it's the type strain of Pseudomonas syringae, okay, from lilac. It causes blights of cucurbits, blights of sugar beets, all right, for example. So this is one that is really seems to be important. So we did host range testing of like over 20 years, and we put these data together, and I think there's about 135 strains in here. We did host range testing on between 16 and 22 crop species in 4 blocks over 20 years. So because over those years, we changed our orientation, we didn't always test the same number of we didn't always test the exact same plant species. So I couldn't for all strains do a comparative host species, but I can tell you the frequency of plants that were attacked. All right. So here, all these strains are from this haplotype DD1. And so if we blow it up, we can look at the intensity, the percent of susceptible plants out of those 16 to 20 that were crop species that were tested. Here in this group, there are a lot of blacks and reds, strains that attack between 75% and 100% of the plants that we tested, okay, and under lab conditions. All right. So my first question was, I want to survey those, right? I want to see if there's a way I can trace those, right? And so these strains, about 50% of them were pathogenic on over 50% of the plant species tested, but there was a lot of variability in this host range in terms of individual species that were attacked. And here's one block where I can make the comparison. You can see, I can present this block of strains with the phylogenetic tree on the left, and then the plants that they were tested for. And you can see right away that strains that are close to each other on the phylogenetic tree don't necessarily have the same host range. So you go, okay, that's good. What do I do with that? Right? And if I look at this with what we call nested analysis, and we do the test for patterns to look for pathotypes, like modules of strains that are all have the same host range or near host range, and they're distinct from others. There isn't. It's just an overlapping continuum. So it's almost impossible to predict pathotype, host range for any given strain. Okay, it's based on its phylogenetic situation. It's very, very difficult. So so I said, okay, so now let's go back to these strains, and now we have information about clonal lines. Maybe strains that are clones, whole genome, have the same host range, right? Because maybe those differences are due to the differences in their genome, because we're just talking about haplotypes here. So in this part of our diagram, I'm going to show you groups of clones. All right, and these are the clones for, we're going to start with the clones for that DD1 haplotype, whole genome clones. Here we go. Here's one clone. These strains in red are whole genome clonal lines. Okay, of course, there's a small percentage of difference. Look at the difference in their host range. All right, again, another clonal line, number 2 in yellow. Look at the difference in their host range. Another clonal line within this DD1 haplotype in green or greenish brown. Again, they don't do the same thing. Then we have a clonal haplotype from phylogroup 2D, and the blue are whole genome clones. They have different host ranges. Well, I got to this point in my research and I said, I'm not looking for markers of host range. There aren't any. This is where I just really got frustrated, okay? Because even clonal lines, okay, they're not, and of course the sequences aren't 100% identical, but we're talking about 98%, 99% identity. They don't have the same host range. Okay, so the question is, what are the underlying molecular mechanisms to describe this? Okay, there's no correlation with effector repertoire, okay? And is this modulated by the environment in the same way for all strains? Can these be targeted by surveillance? I mean, my feeling is not yet. And just for information, I have a student right now co-training with Elodie Vandel at the University of Verona in Italy, where she's looking at how important temperature is for the speed in which Pseudomonas syringae induces the hypersensitive response in Arabidopsis. So there's differences in sensitivity to temperature even that can make or break it. And yeah, so that's a important thing. So there's fine details here that make a big difference in the outcome. To go a little bit further, when we saw this overlapping continuum idea in Pseudomonas syringae, my feeling was, oh yeah, everybody's going to say it's just Pseudomonas syringae, you know, it's just unique. But some colleagues gave us data sets for a dozen host pathogen interactions that they had themselves. So Pseudomonas syringae on apricot, Puccinia hortii on barley, there's a nematode, okay, there's viruses, other fungi, and we analyze the host range patterns for these, and it's all the same. They're all nested. There's an overlapping continuum. There's no statistically significant pathotypes, modules of pathotypes, which led us to make this title, The Quasi-Universality of Nestedness in the Structure of Quantitative Plant-Parasite Interactions. So this was the subject of the talk that I gave at ICPP. What does this imply for the concept of pathotype? So I put the big frown there. We can talk about it later, but there aren't pathotypes, okay, pathovirus. Anyway, on to the next subject. So if, for example, when we saw that haplotype DD1 was really abundant, if we targeted that for surveillance, we'd be sounding the alarm bell all the time in the Durance, and unnecessarily, because there are, in fact, very few epidemics caused by Pseudomonas syringae in the Durance River Basin. There are some on apricot and lettuce, but there's nothing extraordinary, okay? And so my feeling is that our observation is like times zero. We observe this under given environmental conditions of water chemistry, ambient temperature, land use, and cultivars. And so perhaps the key thing to be watching, and I'll talk about this later, is change of these situations. maybe this situation here brings out the natural processes that keep Pseudomonas syringae as a saprophyte and an epiphyte, and no, it keeps it down. Most everybody's happy. Not too much fertilization, no over stress of crops. And so maybe these conditions are right for limiting disease. Okay. So maybe we then should survey for deviations of these conditions, significant changes in land use, introduction of new crops, changes in water temperature, water chemistry, water temperature, other things. That might be the key, rather than just surveying for the pathogen, because the pathogen's always there. Okay, so what to survey when and where? This is a little bit of my opinion. So this is my personal opinion based on 40 years of research in ecology and epidemiology. There's some use for this concept of pathotypes, okay? Very specifically, if the full genetic diversity of the pathogen is known and limited, take something like mycobacterium that causes tuberculosis, okay? Tiny little genome, we can know it's, so something like that, if we have in plant pathology, something with a small genome where we know it's really restrained and there's not much genetic diversity, then we can maybe survey and using the pathotypes and say, okay, there's some variants we need to watch out. We would also need to know the full pathogenic potential of a pathogen. The phone in my house is ringing. Sometimes that cuts my internet, but anyway, just if it does, I'll get back on. So we could use pathivars or pathotypes if all the genetic potential, the pathogenic potential and reservoirs are known, okay? Because we have to take into account things coming from elsewhere. It might be useful in the case of production of vegetatively propagated plants or certified seed when the exchange is via the planting material, okay? Or when there are obligatory quarantine measures that specifically restrict what the pathovar is, but with the knowledge of its full genetic diversity, which isn't always the case. Otherwise, I think disease surveillance needs to be transformed into mapping risk potential, okay? Where you take into account the pathogenic potential of the full metapopulation, not just the population causing the disease, but in all of its reservoirs, the mechanisms that underlie the variability of this potential. We need to know how what the external reservoirs are and estimations of their connectedness to crops. So if the connectedness is via irrigation water from a river, okay, that needs to be taken into account. And then we need to think about climate and land use variability over a scale of time in which you're going to implement the control measures, okay? So we need to think about that. If you're going to put into place control in an orchard, that might take a really long time. And so you need to think about all the things that are going to be changing in terms of climate and land use during that time. So that's what surveillance probably should take into account in the future. And that we need to get rid of, this disease triangle, in a way, it's a handicap, okay? Because it's static. It says there are three actors: plant, pathogen, and the environment. But disease is not just the actors, it's a process, okay? And that's what we need to be surveilling, the components of the movement of this process, all right? And today our surveillance is really oriented around the pathogen, specific environmental conditions, and the sensitivity of the plant. But we forget about the process, and it's not an object, all right? And this process has stochastic aspects, as well as mechanistic or deterministic aspects. And I don't think we've got our head around the fact that there are random processes that go into making disease, all right? And it's very uncomfortable. Most of us don't like that because it's difficult to predict. But there, if you know that there's stochasticity in lots of traits in the environment, and we need to take that variability and the stochasticness of it into account. So we'll finish on a little trip to Iceland. We'll go back to Iceland. Are you ready? So here is something to think about for the future in terms of how diseases emerge and how we can watch this and how we can survey. I think that Iceland, well, it represents a geographic region at the dawn of agriculture, where climate and land use changes are expected. If you go to Iceland today, almost nothing that you eat comes from Iceland, but little by little, they're growing, they're trying to grow crops, they're trying to figure out what they can do, and that with climate change, it's going to happen. So, you know, what would be the scenario of invasion and colonization by Pseudomonas syringae? And what would be the scenario for other pathogens or insects? And will it happen as agriculture intensifies? You know, this is a place where we could ask this question, all right? So let's go back to that graph where we had these monophyletic groups of Pseudomonas syringae that were like nothing else in Pseudomonas syringae. How did this happen? And so, of course, I mean, this is my speculation. So we're going to look at these phylogenetic groups. There's about 17 of them. And instead of representing them on this phylogenetic tree, I'm going to line them up in a graph. So here they are on this graph, L1, 1, 1, 2, etc. And I was able to estimate the time of divergence of each of these groups. And that was thanks to a tool, a bioinformatics tool, and the fact that 3 other researchers had published dates of divergence of certain groups of Pseudomonas syringae. So that were my markers in my tree, and I was able to date these. So this is the length. So phylogroups 1 and 2 on the bottom are your main pathogens. 4 causes diseases to trees. 7 is Pseudomonas syringae. 10 and 13 tend to be ones that don't cause disease and you find only in the environment, okay? And so I put them on a time scale of in log base years. So 10 to the 7th is 1 with 7 zeros after it. So it's 10 million years, okay? So, and on it, I put on this time frame some major geological events. The oldest plant fossils in Iceland, the last glacial maximum, and then when that big glacier that covered North America melted. So here's the scenario. Iceland is only 50 million years old. Okay, it emerged from the sea. It's not a volcanic, it wasn't a volcanic explosion, just merged from the tectonic plates of the sea. And so already you can see that none of the monophyletic groups of Pseudomonas syringae that for their age I estimated are older than 50 million years. So this is already the first part of the coherence. They're not older than the island itself. All right, that's really interesting. Okay, then there was the last glacial maximum, and what people have said about it was that no plant life survived. Nothing. There were not refugia on Iceland. So eradication of all plant life. Pseudomonas syringae itself could have survived in ice and under glaciers. And I called my colleagues who work on viability in glacial ice, and they said, oh, 20,000 years? That's a vacation, no problem. Many bacteria can do that. So it's a real possibility that Pseudomonas syringae could have survived that period in ice and under glaciers, okay? So then, when when there was a deglaciation, that caused, it was actually very rapid, about 10,000 years ago, and it caused a nice movement of water, a faster movement of water in the North Sea around Iceland, and it brought with it flotsam of plants, mostly from Norway and Scotland. And today, 80% of the plants in Iceland have counterparts in Norway. I think, yeah, 80%, and about 65% have counterparts in Scotland. Okay, so those plants from those places recolonized. So maybe they had Pseudomonas syringae on them, and they came. Okay, they came over, but they're still separate. And then since then they've been evolving. And so the real clue here is the fact that the jet stream does not go to the places we sampled from. The jet stream only touches the lower part of Iceland. So when the volcano, the Eyjafjallajökull, whatever it's called, blew up in 2010, and we got all that dust in Europe, that was because it's really on the most southern part of Iceland. It's a very odd case. But mostly their Pseudomonas syringae isn't transported there because the main air masses that move Pseudomonas syringae around the planet just don't go far north enough. Though most of Iceland is in the Arctic Circle of air movement. And so that's how they stayed away. They stayed apart. So here's my last slide. in thinking about all this, what questions would you ask about the evolution of Pseudomonas syringae or about other pathogens? If you could have a situation like this to see the beginning of. So I think that's it. With that, I'd say thank you for your attention, and I'm really excited to hear your questions.
The question is about the NOAA database. Do you look at special permission to access it?
No. It's open access, free. High split, type high split. There you go. You got it. Yeah, it's open. I mean, we downloaded tons of data and we really didn't, I think maybe we told them, but no, they don't care. But let me say something about this. In 2020, no, 2016, yeah, when Trump won the election, before he took office, I got a carpal tunnel syndrome downloading data because there was a menace that all these places were going to be closed to environmental scientists. And so, but in fear that these databases were going to be closed, I downloaded like crazy and got myself a carpal tunnel syndrome. So, so no, it's free. It's free.
Hi, Cindy, it's Lucy here. That was a really interesting talk, and I had an opportunity to also listen to Marianne Barney. I work on Bacterium.
Okay.
So I was fascinated seeing what you have presented and what she and her student presented at ICPP. It's really interesting that you couldn't find any genes that would indicate a host range, right? I was wondering, in terms of the isolates that you find, were they all pathogenic on plants, or did you find some that were not pathogenic? For example, if you isolated from water, would they all be able to infect some plants?
Yeah,
or
okay.
did you find some isolates that could not infect at all?
Okay, all habitats, all substrates that we isolated from have strains that can cause disease on plants. Okay, every substrate has some strains that can cause disease on plants in artificial inoculations. Okay, but there are, when we started this work, we have found strains that have type 3 secretion systems that are damaged. And when you sequence them, you can see it. And when you do the hypersensitive reaction, you don't get a result. But I mean, they're rare, but they do exist.
So I was wondering if there are some Pseudomonas syringae that are, for example, that survive purely by living in water. And if you find genes that would indicate how they survive in that particular environment. Yeah.
So what's intriguing is that phylogroup 10, it is the one that we find not so often associated with plants, and it's never been known, it's never been declared as a cause of any disease that I know of. Maybe there's one odd case. It is mostly in water, fresh water, of course, and we don't, I mean, we're trying to understand, but if you look at the genomes are nearly identical in composition to phylogroup 2. They have the syringomycin, they have effectors, they're isonucleation active, they have the non-ribosomal peptide synthetases. And so the question is, what is the difference between these two groups that otherwise genomically are so similar? So that's one of the outstanding enigmas.
Very interesting.
It could boil down to something really small, okay? Really small, like maybe they overproduce lipopeptides or something. Or, you know, and that where they get in with a plant, they just like are screaming. So the plant says, it's over, right? So we never see disease because when it happens, it's just catastrophic. You know, it could be that.
Yeah, I guess it goes back to what we said at ICPP, that we need to sample much more than just looking at things that we find in the plant environment.
Oh my gosh, yes. Okay, completely. Most of our collections of strains are clones from epidemics, right?
Thank you.
Thank you for the question.
I'm then going to ask Bernard to close the session. So Bernard is the director of PABI and he's sitting in the audience. Can she see you?
Yeah, I can
Yes.
put my camera on. And I hope, Cindy, I hope you can hear me.
Yes.
Right. Yeah, Cindy, thank you very, very much for a beautiful story that you told, and challenging many of our assumptions as you've done for a long time. Also enjoyed the interactions at ICPP, but it really is fascinating work that's really making us think in many different ways. I'm thinking about so many of the systems that we're working on, and where we really maybe need to challenge many of the assumptions that we make that we're making about host interactions and how epidemiology about many of the systems, and really need to be addressing it in this holistic manner that you've explained to it. And apart from the amazing work, you told a beautiful story. So thank you very much for spending the time with us this afternoon and challenging us, making us think. We look forward to continuing the interaction and many more interactions that lies ahead. So all the best for your work, and thank you very much for this afternoon.
Yeah, thank you for the remark. Now, I want to say something, because this idea about asking challenging questions and making people think. I'm going to myself like, what is it? Why what's the key? And sometimes I think the key is trying to put plant microbe interactions, for example, in the perspective of human society and diplomacy. And there's all sorts of interactions. We have people, we have freeloaders, and you have aggressive ones, and you have negotiators, and you got ones that are fasting, and ones that eat a little bit, or those that only have the paleo ones, and the vegetarians, and and I would say that the same diversity is true for microbes and plants. There's all the different types of interactions, and it's not to make light of the importance of having a scientific concept, but it could help your creativity to put it in the context of something we know really well. Everybody's hungry, and how do I get my food?
Yeah. No, absolutely. No, thank you very much for that. I mean, I especially also enjoyed the discussion around the disease triangle. And I think, you remember from the discussions at ICPP, I think these are really, as you're saying now, really important and valuable concepts, the things that we must be careful that they have pitfalls in how they make us think in an oversimplistic manner sometimes.
Yeah.
So thank you again.
You're welcome. Thank you. Other questions, please. Come on. There's lots of young people in the audience, I know, and they're afraid. But it's so easy on the internet to ask a question.
Are there any last questions? Let's see.
So I have one. So recently we've been getting a lot of disease notes on very odd bacteria causing diseases in plants, like Klebsiella pneumoniae. What do you think about that?
So they're causing disease on the plant?
Yes.
And you can reproduce this experimentally?
So they say. And it's not only that, there was 106 reports in the last 10 years on unusual bacteria causing diseases in plants.
Well, okay, there's several things. One, people are asking new questions they've never asked before. So, right, that's one explanation. Two, I would think that it would be important to have the strains and look at their genomes to see if these strains are any different than the Klebsiella that classically is associated with soils or human lungs or whatever, to see if there's some evolution of these strains. Okay, and then the context of the plants is really important. Are these plants that are new crops that are stressed, that are not being produced correctly under odd conditions, too much humidity, like lactobacillus for people who have HIV, right? I mean, some strange things where it's an unusual immune system, immune context.
It's just interesting that there's just an increasing number of reports of things like micrococcus and really odd bacteria, so-called causing diseases in plants.
Yeah, and it might be the plants have a poor microbiome.
But they always state that the severity is quite high, between 60 and 65%
Wow, well, I would think it would be important to validate these. I mean, this could be something that plant pathology could say, well, okay, we need to know what's going on here. Are these really new? Or are these just people looking for something that's always been around? Or, you know, I think it would be something, aren't you on the bacteriology committee?
Yes, yes.
Yeah.
I am. Yeah, I just find them hard to review.
Cindy, thank you again for this afternoon and for stimulating us, and Marcus and Merriman. We think about Merriman who's been involved in the arrangements, who's sick at the moment, and for everybody who's joined, I wish you all the best.
Okay, thank you very much.
Thank you very much.
Okay, have a nice afternoon.
You too.
Bye.